coordinate physico and biochemical changes during seed storage. Physical and biochemical changes during seed storage. This important topic. First, let's see the deterioration types. And my voice audible?
Yes, sir.
Right. Deterioration types. And another important topic in this coordinate, seed deterioration symptoms. This both very important. And also testings also. Right. So physical and biochemical changes. First, let's take changes here. Worklet. One minute. Okay. Is it visible? Is it audible to you?
Yes, sir.
Right. Actually, sorry for inconvenience. Due to the some tech. So far the give credential, just listen. After that, I can share. So in deterioration, first in this topic, deterioration types. Weathering. First of all, the first one, weathering. One of the type is weathering.
Sir, your voice is not audible, sir.
Now clear. Hello? Is it audible?
Yes, sir.
Right. So first of all, what is deterioration first? What is general term of deterioration?
Rusting or decomposing.
So some related to that, right? Deterioration means, so actually the deterioration means
Decaying.
destroy something destroy.
Yes.
The quality, that means the poor quality appear as a poor quality. That is deterioration. But here we consider the seed deterioration. So what happened here? The decrease the percentage of germination in the seed deterioration. First in this concept, first germination quantity decrease, while those seeds that germinate produce weak seedling. There are two. First of all, actually in deterioration seeds, first the percentage of the germination decrease. Even any germination occur, the weak, poor seedlings are produced. Right? So now how the deterioration takes place, let's see here. By weathering, one of the first main one is weathering. So during the weathering, the seed deterioration, quality, vigor, and viability occurs due to the high relative humidity and high temperature during the post-maturation and pre-harvesting period in the field. So this weathering may be occurs. First of all, the reason for the weathering, the high relative humidity and high temperature. These two aspects mainly happens during the post maturation. That means after maturation of seed, of okay, there is no interruption during the maturation. But after maturation, these seeds, that means the healthy seed, when you apply, that means when you keep in the high relative humidity and high temperature, what happened during this period, due to the high humidity and high temperature, the deterioration takes place in the form of weathering. You know, in general, the what happened, first of all, after seed storage inside the bag, if the bags keep in the godams, in the godam premises, the any humidity, that means the water or high humidity the formed, what happened? The seeds are the viable, the seeds are be healthy in the water condition. Hello? Right, just wait. Right. Can you see whiteboard? Hello?
Yes, sir.
Right. In deterioration, so change color. In deterioration types, first one, weathering. So this weathering mainly occurs by humidity, high humidity, as well as high temperature also. High temperature. So this humidity, high temperature mainly occurs during the post maturation and pre-harvesting period. Pre-harvesting period. What is pre-harvesting period? Pre-harvesting period in field. Pre-harvesting means when you seedling, when you take the seeds.
Before harvesting, sir.
What is what? Okay, right. What is harvesting here?
Taking the seed.
What? Harvest. The process of gathering crops. Gathering crops means actually after you seedling, after you plant, after put a seed, it produce the seedling. After you give the water, sufficient adequate water, and also it give the fertilizers, whatever it may be requires, all you provide. Finally, it gives the yielding, and now it is ready to take the crop. During all this period called as harvesting. That means the pre and after finally after when the yielding comes, you cut the crops and you store the yielding. So when you cut the seeds, sorry, when you cut the crop, you cut the crop after maturation, right? After maturation. So here these two aspect, one is relative humidity, high temperature. These two occurs the post-maturation and pre-harvesting period. Pre-harvesting period means before maturation of crop. Now to be matured during the harvesting period. After harvesting, now the matured crop, next stage. So even during the harvesting, pre-harvesting period, after harvesting, after maturation seed, in the both situation, any time, if you affect with two aspects such as humidity and temperature, automatically what happen? Deterioration takes place. This is called weathering. This is called weathering, and the weathering occurs in the period between the attainments of physiological maturity. Where it is? Okay. Physiological maturity till harvesting in the field. So this deterioration caused by the weathering is directly related to the seed exposure to adverse condition. What are the adverse condition? Already told you. What are the adverse condition? Hello? What are the adverse conditions? Here the adverse conditions are high humidity and temperature. These are the adverse condition, right? The exposures of hot and humidity conditions, rainfall, photo period after ripening or pre-harvesting factor cause the seed quality, the loss of following physical maturity, isn't it? So if you expose the matured seed after harvesting matured seed, if you expose to the humidity condition, rainfall, high light, temperature, now these are all factors caused to the deterioration. As a deterioration, the seed quality lost the following physical maturity. Among all these factors, influence of moisture on seed during repairing appears to exert the major influence on pre-deposition to weathering. So anyway, the adverse environmental condition during seed filling and maturation result in forced seed maturation, which is associated with low yields. Automatically what happen? During deterioration, due to the deterioration or due to the weathering, the crop yielding or crop productivity would be loss, would be decrease. Right? Hello?
Yes, sir.
Right. The timely harvesting avoids prolonged exposure to moisture and is the best means of avoiding weathering. So you knew what are the reason for weathering. So if you avoid the weathering, what you do? So what are the reason for weathering? Hello? Weathering seed deterioration. Weathering and also seed deteriorations, the reasons. Already told you the reasons are high humidity, high temperature. If you avoid, do you want to avoid this? The timely harvesting avoids the prolonged exposure to moisture, which is the best method to avoiding weathering. And also the harvest and post-harvest deterioration seed quality is highly affected by harvesting and handling methods. And also you should harvest the crop in proper time. You do not harvest before and do not harvest after a long time. After maturity stage, long time also do not harvest. So these two factors are major one to avoid the weathering process. In this process, let's see the deterioration in post-harvest. How then post-harvest, how deterioration takes place? So post-harvest means you threshing, processing machinery, seed collection, handling and transporting, drying, during storage. After drying, it is stored in bags. So they are all aspect comes under the post harvesting. During this any aspect, that means comprises threshing. Threshing means you collected the crops. Thus threshing, processing machinery. The processing machinery means from the crop you separated the seed or fruits, whatever it may be, separated, and also the clean. While you separated, apart from the required seeds or fruits, the waste material also we should remove it, and that means keep it clean. What you require, that only you select. That is processing a machinery. So anyway, after cleaning, they collected the seed and handling means here the packed in bag or whatever it may be, polythene, any type of, and also transporting one place to another place. After transporting, before seedling, that means uh, before giving to crop, you should dry and again store in our godams. In this any, for example, you during the threshing or machinery process, seed collection, in any aspect, here there is a chance to damage of the seed. Maybe the mechanical damages mainly occurs during processing. Maybe mechanical injuries occurs during transportation. Mechanical injuries occur during storage. Also, mechanical injuries occur. Such damage may result in physical damage or fracturing of seed pods. Physical damage means such a that is broke the seed coat. So what happen if the seed coat the break, any physical damage occur, that is vulnerable to fungal attack. What happen if the fungal attack the seed? Hello? If the fungal
We
attack the
can't
seed,
sow that seed, sir.
right? So that is happening during the post harvesting. The how the deterioration takes place, storage of seed. The storage of the seed mainly genetically regulated and is influenced by the quality of seed at the time of storage, pre-storage history of seeds. So here the storage of seeds mainly depend on genetical background of the seed and also environment factors also. For example, during storage period, any untoward characters present inside the gene, then only even you properly stored, they lost the germination. The seeds can get the loss of germination. The another one is environment factors means the external. That means in storage area, the ensure there is no water and the room temperature, it has the room temperature. There is no low and high. If low temperature, it is not optimum. High temperature also not optimum. So these are very, very important during for the avoiding the deterioration of the seed. Okay. So this is about seed deterioration types, mainly focus on weathering. So weathering occurs by high humidity, high temperature. The high humidity, high temperature mainly occurs during maturation or after pre-harvesting period also, right? So another next important one, the storage deterioration symptoms. Okay, you have learned how the deterioration takes place. Now the symptoms. What type of symptoms appear in deterioration? Uh, let's share the board. whiteboard. Are you able to see the board?
Yes, sir.
Right.
So yesterday you have completed with two chapters.
Yesterday, yes. What happened here? Marker, I can't take marker. Where the marker here? Right. Sorry. Right. Seed deterioration. Are you able to see? Hello?
Yes, sir.
Seed deterioration symptom. First of all, some changes are morphological appear. So let's see the morphological changes. Morphological. Morphological means next one.
The body, outer
Right. External.
area of the seed, external.
Body. Body morphological, external morphology, internal morphology, both. So you're only given the morphology changes. So in morphological changes, and next one, ultra structure. First, let's complete morphological changes. In morphological changes, the mainly symptoms appear on seed coat. That is what one of the morphological structure of the seed. How the symptoms appear in seed coat? Actually, let's change color. The seed coat color are apparently appearing very dull in color due to the oxidative reaction in the seed coat that are enhanced under the condition of high temperature and relative humidity. This is common. From beginning, already I'm telling you the two major aspects to deterioration. One is high temperature and one is relative humidity, high relatively humidity. But now we are discussing how the deterioration symptoms appear. So in deterioration symptoms appear in the seed coat. In seed coat, the pale color, that means seed coat completely pale in color, appeared in pale color or dull color. This is done by oxidative reaction in seed coat. The reason for that, high temperature and relative humidity. And another one is ultrastructural changes. If you see internally, by electron microscope examination, to broad patterns of coalescence of lipids bodies and plasma lemma extracted from the cell wall related with deterioration have been observed. Both of these events influence cell membrane integrity. In morphological, first one, externally seed coat appearance. If you see the internal, the plasma lemma or plasma membrane, the plasma membrane deterioration takes place. The loss of integrity of cell membrane. So this is happens in ultrastructure changes. Let's see how the cell membrane changes here. See deterioration. Mainly the cellular constituent decrease, which was attributed to cell membrane.
Sir,
This
in the ultrastructural changes, can you
hmm,
show how it varies?
in ultrastructural changes,
How the seed is deteriorates, sir?
only by in ultrastructure only these symptoms appear in plasma membrane, plasma lemma. That means here the plasma lemma extraction from the cell wall related with deterioration have been observed. What I'm saying, in first step we can observe the symptoms in seed coat. So the plasma lemma, the core essence of lipid bodies and plasma lemma extraction from the cell wall. Do you know where the plasma lemma? For example, this is the seed. For example, this is the seed coat. In this seed coat, it is made up of number of cells. The seed coat or seed, this is made up of number of cells. If we take one cell, the cell covered by cell wall. After cell wall, the next is plasma membrane or plasma lemma. So in plasma lemma, in second layer, plasma lemma, what happen if we separate the core essence of lipids bodies? That means on this plasma lemma, some of the lipids bodies appeared. For example, if you separate the plasma membrane, phospholipids, proteins, both are present. Phospholipids and this is the structure of plasma lemma, plasma membrane. But when you affect with high temperature and high humidity, here some of the coal essence that in black color spots and also lipid bodies. This is completely made up of some lipid bodies. When you observe in microscope, these lipid bodies appeared in plasma lemma. Based on the symptoms, based on the lipid body symptoms, you can understand this seed undergoes to deterioration. First of all, actually, we make sure the seeds are deteriorated by seed coat symptoms. And second, internally, when we observe the not only normal microscope, electron microscope, when you separated the cell, when you separated the plasma membrane, then only the symptoms appear. In this both situation, we can understand the seed get deteriorated, right? And in clearly next given the cell membrane changes. In cell membrane changes, the capability to retain the cellular constitution decreased, which was attributed to cell membrane disruption associated with the loss of membrane phospholipids. Already told you the plasma membrane made up of phospholipids and proteins. But this integrity, the structural integrity of plasma membrane loss due to the composition of lipid bodies. Some consequences of membrane damage includes break. What happened? What happened if the integrity loss? Maybe the plasma membrane breaks and its contraction from the cell wall. The fragmented endoplasmic reticulum lacking of polyribosomes, lack of dictyosomes, monosomes, attribute distressed. This much no need. Just remember one word. In cell membrane changes, mainly the loss of phospholipids. That means association with loss of plasma membranes happened. What happened if the loss of phospholipids loss the integrity of the structure? That means there is no proper structure to plasma membrane. Why? Because actually the plasma membrane made up of phospholipids and proteins. What if the phospholipids loss? Is there any structural? Is there any structural to the plasma membrane, a structural support to the plasma membrane. If the loss of
No, the
phospholipids,
integrity will lost, sir.
like, and also inside the cell organelles also properly not arranged. Inside plasma membrane means if you separate the plasma membrane, inside dictyosomes are there, monosomes are there. They are also not properly arranged, right? So now the next one is loss of enzyme activity and reduced respiration. Enzyme activity, which enzyme? First of all, remember which enzyme is involved in respiration. Mainly the amylase enzyme released during the germination. So let's see the how the enzyme loss and how the respiration reduced. For example, it actually given one test. The test name is tetrazolium test. The most sensitive test for measuring early seed deterioration. Tetrazolium test. Remember it. Tetrazolium test recognized for the germination percentage and also recognized for the respiration. Based on the tetrazolium test, we should calculate the deterioration percentage. That means how many seeds are under deterioration. For example, in tetrazolium test, what you find out in tetrazolium test, you can find out the amylase enzyme activity. If there is no amylase enzyme, that means I am asking a simple question. In deteriorated seeds, is it enough uh, amylase enzyme in that seed? First, let me complete in general. Just wait 1 minute. Correct. Seed due to amylase amylase enzyme in presence of amylase enzyme, seeds germinated. Seed germinate. So there is no deterioration if properly seedlings happen, properly seedling produced. But due to some reasons, the loss, the enzyme, which enzyme loss? Mainly the amylase enzyme loss, and also other oxidase enzymes, catalases, peroxidase, cytochrome oxidase. These are all respiratory enzymes. They are all correlated with the seed deterioration. In seed, if these enzymes not produced properly, now the seed undergoes to deterioration. As seed deteriorated, respiration becomes gradually pathetic, evenly leads to the loss of germination. Right? So how you find out? How you find out inside the seed whether the amylase enzyme or other respiratory enzyme present or not? You can find out by tetrazolium test. Tetrazolium test already it has learnt in during laboratory. Right? Is it clear? So that means what I'm saying here, So deterioration symptoms. The first symptom, morphological seed coat. The seed coat changes appear due to the plasma membrane of the seed coat cell. Later, the another symptoms is due to the high temperature and high humidity, here the enzyme activity loss. That means how the germination takes place? The germination takes place by some of the oxidases, respiratory oxidases enzymes. But during this high temperature, high humidity, the enzyme activity loss, that means amylase enzyme loss, this is responsible for respiration. Due to the respiration, what happens? Germination takes place. But due to the effect of this loss of amylase, there is no respiration, there is no germination, isn't it? Hello?
Yes,
Hello?
sir.
Right. So these, the increased seed layers, they no need. Only the seed deterioration symptom, this much enough. Morphological changes, seed coat, ultrastructural changes in plasma membrane, and loss of enzyme activity. These were enough. And remember, in cell membrane change, don't get confused in cell membrane changes. In cell membrane changes, mainly the changes appear in phospholipid. Phospholipids. That means the lipids body patches formed as in coalescence. So these symptoms appear in plasma membrane. So these are the deterioration symptoms. Is this clear? Next one, some testing. Testing not important. Already here the tetrazoline test you must have learnt in the laboratory. So and these are all belongs to the just practicals. No need for theory. So anyway, just read it. Germination test, tetrazoline test, electrical conductivity test, vital test, and enzyme activity test. Right. So anyway, remember in third unit mainly one is deterioration types and it the seed deterioration mechanism, no, not important one, and seed deterioration symptoms. Next one, seed viability, fourth unit, block 2, fourth unit, seed viability and genetic erosion. Very, very important. This both. Right. Seed viability and genetic erosion. First of all, what is seed viability?
Seed germinating capacity.
Right, that's it. Where is marker? So seed viability means seed capable of germination, right? Right, okay. Now the factors affecting the seed viability, one topic, and genetic erosion. In this most important one is genetic erosion. Factors affecting, this is general, factors affecting for seed viability. First, let's see the affecting seed viability. Factors. Factors for seed viability. So here the given one by one. Cultivars and harvest variability. That is first one. Initial viability, pre-harvest, post-harvest. First, before you should aware what is viable. So as you said, the viability means the seeds is capable of germinating and producing normal seedling. Now let's see the factors affecting seed viability. What are the factors? First of all, cultivars. Cultivar, one factor, cultivars and harvest variability. Different cultivars and harvest of species may be show different viability. Different viability characteristics under the same storage condition. The significant difference in viability period exists between the genotype within the species. Actually, before genotype, here the what I'm saying here, the cultivars and harvest variability. For example, if you take the resinous communicus, castor oil, and you can take the cotton, you can take the rice. Can you use the same cultivars? Can you follow the same harvest method? These all crops. Hello?
No, sir.
No, that means the different cultivars and harvest of species may be show different variability. And also another factor for the viability is genes. That is another factor. And initial viability. It has been established that healthy, well-matured seed with high initial germination capacity will retain viability longer period, and then that seeds having low initial germination capacity. That means viability. Initial viability, that means the initial viability generally occurs in genetically. Already genetically strong, if any untoward actions appeared during the harvesting, pre-harvesting, post-harvesting, seed storage process, even any untoward actions happen, there is no changes in viability. That is initial viability and pre-harvest and post-harvest condition. So we make sure this harvesting process, we take the crop in proper time. After harvesting, we storage, we processing, threshold, processing, also proper time. This is also necessity for viability. And actually, some of the things missing here. Mechanical injury, tetragonium, these are all actually no needs. Sir, improper, I think. So anyway, so next aspect is mechanical injuries. If any mechanical injuries happen to the seed, what happen? Seed viability increase or decrease? Any mechanical injuries. These mechanical injuries may be happen during harvesting period, after processing, and during storage, during transportation, in any time. These mechanical injuries happen. Just tell me the simple. In any mechanical injuries happen, the viability increase or
Viability
decrease?
will decrease, sir.
Will viability is decrease, right? Now the next one, tetrajolium test for seed viability and vigor. Just now we discussed the tetrajolium test, the tetrajolium test for germination of the seed. This germination of seeds occur during respiration. What happened actually during respiration? During respiration, ATP enzyme produced. With help of the ATP enzyme, this energy utilized for the germination of seed. This is done by amylase enzyme, right? So now let's see the tetrajolium test viability. Okay, here the germination test is the best indication of the potential of the seed lot emergence. Altered this, whatever it may be given, just remember one word. This test for germination of seed. So anyway, it takes from days to weeks and some cases even months to complete this test. Because actually here we not only taken the one field crop, we will have to observe the different crops. That's why taken more time. Out of which tetrajolium test, commonly known as TJ test for seed viability, have therefore we have viable means already. He told you the capable of germination, right? Hello?
Yes, sir.
Right. So the test is very useful for processing and handling, storing, marketing of the seed. So if you after test, it gives the positive. That means inside the seed amylase enzyme present. Now immediately it is ready to buy. It is immediately ready to sell. That immediately you processing and handling, storing, marketing immediately. Because if you do any delay, there is a chance to imbalance in enzyme activity. During the if any imbalance happen, automatically the viability decrease. That's why after test immediately you marketing it. And let's see the advantages of tetrazolium test and disadvantages of tetrazolium test. First of all, the advantages, the seed viability can be estimated. That is the first advantage. From beginning, we are discussing that. And these techniques can be used even the both dormant seeds or where the seedling growth is extremely slow. Of course, tetrazolium test only the recognition of germinating. At the same time, germinating means viable seeds. If you take the some of the different crop of the seeds, in the seeds, can you find out only germinating seeds? Hello? Can you find out through the tetrazolium test only find out the capable seeds? Can't you find out incapable seeds? That means dormant seeds. Can't you find out tetrazolium? Tetrazolium test positive, that is active seeds, viable seeds. Negative, automatically dormant seeds. So this tetrazolium test not only for viable seeds and also non-viable seeds also it is useful to determining non-viable also. For example, what happened during this period? We selected the seed first. The selected seeds soaked in water to enter the day after seedling comes out, or even that is not a matter. After soaking in entire the night, we cut the seed longitudinally and keep it in tetrazolium after almost 2 hours, 2 to 3 hours. If you observe, the pink color stains appear. If the pink color stain appear, ML is enzyme present and it is viable seeds. There is no pink color, they are non-viable seeds. Right? The since dicot seeds need not be damaged during the tetrazolium test. So actually, if we use the chemicals, this chemical there is a chance to damage the embryo. But the dicot seeds, especially dicot seeds, not damaged during tetrazolium test. The same seeds could be utilized for further investigation also. Now let's see what are the disadvantages. The best disadvantages is the evaluation is not easy. After applying staining tetrazolium, the evaluation is not easy to determine the seed viability. It is very difficult. That is one disadvantage. And the total work involved in that test requires greater number of man-hours. This does the germination test. Of course, this, for example, if you want to do some limited seeds, okay. But at a time, if you want to see the uncountable, that means the huge number of seeds, it requires the not only greater numbers and also time and also instruments also. So this is disadvantages. And this test alone is unable to differentiate between the dormant and non-dormant seeds. Already in first point given, somewhat difficult to determine. Is that somewhat determined difficult not only for dormant and non-dormant seeds also. Not only viable, non-viable also. And it is not possible to detect presence of seeds, microorganisms harmful germinating. Even here, the only you focusing on whether the seed viable or not. In tetrazolium, especially tetrazolium test for these seeds, whether viable or non-viable. But you can't determine with it infected seeds or non-infected seed. That is another disadvantages. That means, of course, you after tetrazolium test, you give a certificate. This is the good. Can you give the certificate after only tetrazolium test as it a good seed? Can you give? Hello, we can't give because in tetrazolium test only the viable germination, only enzyme activity we find out, but not the pathogen, but not the harmful activity, harmful pathogens. And another one, injuries caused by seeds by microorganisms, fungicides, and repeated the same point. Right? Is it clear? So this is the tetrazolium test advantages and disadvantages. Of course, this is the somewhat dry topic, but keep focus on it. It's not explanatory when we compare with the core. Core we can explain with help of diagrams, but it is completely dry. It is completely theoretical. That's why just listen. That means just develop the concept. Means, for example, here first of all, what is seed viable? Definition. What are the effectors? Headings. The effector heading cultivars, initial viability, pre-harvest, post-harvest, mechanical injuries. And have you find out this viable seeds by tetrazolium test? That's it. Advantages and disadvantages. This much enough. And another important one is genetic erosion. This is very, very important for long essay, long question. Genetic erosion. Already time at
So tomorrow we are starting from genetic erosion, sir.
definition. Just of all, just
Genetic erosion.
erosion. What is soil? Do you know the soil erosion? Whenever you hear this word erosion,
No, sir.
simple.
I know, sir.
You don't know about erosion. Erosion simple.
I know, sir.
Loss.
Erosion.
Tell me what is that erosion?
Loss only, sir.
Loss of something. For example, soil erosion. Soil erosion means what loss here? Soil fertility loss. The
Yes.
fertile layer of the soil loss. That is called soil erosion. Genetic erosion means what loss here?
Genes.
Genes loss.
Like,
That's it.
not.
And also replaced by new genes. Simple.
Yes, sir.
For example, I would like to just simple thing. One is actually in 1940 before 2000 year, the different type of rice crops are there. Can we use the same crops now?
No, sir.
No, already completely different. That means all type of rice changed.
Yes, sir.
How to change? Actually, before 2000, mainly we apply the only organic fertilizers. But now all completely chemicals and also utilizing hybrids. What about brinjal? What about cotton?
Yes, sir. Golden rice.
Golden rice. Could you find out golden rice in before 2000?
No, sir. I was not there, sir. Before
Of course,
2000.
the same rice also Sona Mysuri, the same rice, but the change, gene change means the same gene loss. There is no same genes in what we used in 1940, 1950. That there is no same plant, no same plant, no same plant means the completely the plant community decrease and it replaced by new community. New community replaced means already what is the existed one? Loss or not?
Yes, sir.
Right. That is about genetic erosion. We will discuss clearly tomorrow. Okay.
Okay, sir.
But
Okay.
remember, genetic erosion very, very important. Definition clear, genetic erosion?
Yes, sir.
Right, right.
Sir, you're having any news about fifth semester result?
Sixth semester?
Fifth semester result, sir.
I can't understand.
Result
Sixth
of fifth
semester?
semester, sir. Fifth semester result, sir.
Result,
It's not
result,
declared
result,
yet.
result, result. Actually, the evaluation process completed. Maybe this month or next month will be released.
Okay, sir. Thank you, sir.
Okay, thank you, thank you.